Good morning to all of you. So today we are going to start the process of translation, and before I start the process of translation, I would like to tell you the structure of TR. Now tRNA or transfer RNA, which is also known as the adapter molecule. This is also known as adapter molecule. And why this is known as adaptive molecule? Because it adapts the genetic code for the formation of, because it adapts to the genetic code for the formation of protein. Now, like structure, as I already told you, clover leaf like structure. So when you see it, how does it appear? It appears to be like this. So it has the 4 sides. This is 5 prime and it goes like this, like this, like this, like this, and ends in 3 prime. And this 3 prime has an OH, and this is the functional group which is present on the 3 prime end. And here the transfer RNA containing the amino acid attaches. Here, this side, it is attachment to the this is meant for attachment to the COOH group of amino acid, which is carried by tRNA. When the tRNA carries the amino acid, the COOH group of amino acid, it attaches to the functional group, which is OH group on the 3 prime. Now this side, this is the ribosome attachment site. This side, ribosome attachment site. You know, ribosomes are very important because these are the protein factories of the cell. So these are very important, and it has pseudouridine. What is present on this side is pseudouridine is present. This is the second site. The third is recognition site. This is the recognition site. I'll write here. With anticodon. This site carries anticodon. That means it will have whatever attaches here, the messenger RNA, it will have anticodon for that. Suppose here we have in messenger RNA, suppose we have U, A, and C. So this transfer RNA will have AUG. It will have A, U, and G, the anticodon for this. And the amino acid it will which this transfer RNA will carry. I always tell you transfer RNA is like a coolie which carries the amino acid. So the amino acid which this site will carry, it will be on the basis of this triplet codon. That means UAC codes for tyrosine. So it will carry the amino acid with tyrosine. This transfer RNA. So it will carry tyrosine. UAC codes for tyrosine. So this transfer RNA will carry tyrosine here, where the amino acid tyrosine will be attached by COH group. If you remember, in 11th class, we have done the structure of amino acid. It has carbon R, CH3, COH, NH2. So this is amino acid. So with this COH, it is attached here with this. So this is how the amino acid is attached, and this amino acid is on the basis of the triplet codon which is present on messenger RNA. This is messenger RNA. So whatever anticodon is present on messenger RNA, that whatever codon is present on triplet codon is present on messenger RNA, that amino acid is brought about by 3' end of the transfer RNA. So UAC codes for tyrosine. So this transfer RNA will bring tyrosine, and the anticodon for this UAC is here on the recognition site of transfer RNA. This is recognition site of transfer RNA. So it is carrying anticodon for this triplet codon. This is U, A, U, C, G. So this carries the anticodon for the triplet codon which is present on messenger RNA. And this, the amino acid which this transfer RNA will carry, it will be the on the basis of the triplet codon which is present on the messenger RNA. So here it is tyrosine, and this tyrosine codes for this UAC codes for tyrosine. That is why it is carrying tyrosine here. The fourth site, which is on the extreme left, this is enzyme site for binding with amino acid transfer RNA synthesis. So this is enzyme site. And it binds with the enzyme amino acyl also, some people say amino acyl tRNA synthetase. This is the enzyme binding site. So here this binds with the enzyme which is aminoacyl tRNA synthetase. This is the complete structure of tRNA which acts as coolie, because now what we have got after transcription, we have got this messenger RNA after transcription. And this messenger RNA, it carries coded message. Coded message, that means in the form of triplet codons. The message is there in messenger RNA, and on the basis of that message which is present in the form of triplet codons on messenger RNA, the protein is synthesized. Now for synthesis of protein, what is required? Protein is basically amino acids, amino acids, amino acids, amino acids. When amino acids are attached by peptide bond, we say it's a protein. It's a protein. It makes a protein when many amino acids are attached by peptide bonds. So now this, according to the message which is present on messenger RNA, the proteins are synthesized. Now for that, amino acids are required. So how the amino acid will come? This transfer RNA will bring the amino acids with it on the 3' end where OH functional group is present. It will carry the amino acids on this end, and this amino acid will be based on the whatever genetic codon or whatever triplet codon is present on messenger RNA. Here on messenger RNA, I have given you one example. UAC is present. So this transfer RNA on its 3' end where functional group OH is present will bring tyrosine, because tyrosine is the amino acid whose triplet codon is UAC. Okay, so this acts as a coolie for bringing the amino acids, and then suppose next we have here AUG. Suppose next triplet codon which we have here is AUG, AUG. G, then what will happen? Another transfer RNA will come here. Okay, I'm just making this much. Another transfer RNA will come here. And this transfer RNA which comes here, it will have the anticodon for this. That means it will have UAC. It will have UAC. Okay, it will have UAC. And then which particular amino acid it will bring? It will bring the amino acid for which AUG codes for, and AUG codes for methionine. So it will bring methionine. So next amino acid will come, and whatever the next information is here, next amino acid will come, and then these peptide bonds will be formed, and finally we will get a protein molecule. Could you understand this, children? Is it clear? Till here, is it clear? Yes. Structure of tRNA. Why it is known as adapter molecule? The presence of anticodon, all the sites of which are present on transfer RNA, the site which binds with COH group of transfer RNA, everything. Is it clear, beta, or any query in this? Yes? Any query in this? Yes, bache? Yes, bache? Any question in this? I cannot get your response. I'm not getting your response. Yes? Any question in this? The structure of tRNA? Okay, Sukhsahaj. Just one response from Sukhsahaj I have got. Any other? Any question regarding this tRNA? If not, I'll move on to the next topic. Is it understood, or shall I repeat this tRNA? You want me to repeat this, or is it understood? I'm not getting response, beta. Is it clear, or shall I repeat this? Except for Sukhsahaj, I didn't get any other answer. Yes, Amit Katoch. Paras, okay, Paras. Always two answers, Paras and Sukhsahaj. Yes, Amit Katoch. Vanshika. Today Chitra is also not there. Arif clear. Vaniti clear. Amit Katoch, is it clear? Amit Katoch. Chitra, fine. Fine. Amit Katoch, can you answer my question? Can you hear me? Okay, fine. Must not be having any problem, that's fine. Let's start with the next beta, the next structure which I want to discuss with you is ribosome. Ribosome is also very important, and ribosomes are known as protein factories of the cells. Protein factories of the cells. So they help in protein synthesis. That is why they form an important machinery of the process of translation, because they help in protein synthesis, because they are the protein factories of the cell. So they help a lot in the process of protein synthesis. Now, the ribosomes in prokaryotes and eukaryotes are different. The ribosomes in prokaryotes and eukaryotes are different. The ribosomes in case of prokaryotes, if it is a prokaryotic cell, the ribosomes will be 70S type. And if it is a eukaryotic cell, the ribosome is present in them is 80S type. What is this S? We have already discussed in class 11. Still, if you remember little, what is S, full form of S? Anybody can tell me? Yes, anybody can tell me? What is this S in case of ribosomes? 70S, 80S, what is this S in case of ribosomes? Anybody can tell me? Yes? Anybody, if anybody remembers, what is this S? No? S is Sweetberg's unit. If you remember, we did in class 11 Sweetberg's unit. The rate at which they settle to the bottom of the centrifuge tube, that is Sweetberg's unit. S in case of ribosomes is Sweetberg's unit. These ribosomes are present in cytoplasm because process of translation takes place in cytoplasm, and the ribosomes are also present in cytoplasm. These ribosomes are present in the form of two subunits, one larger subunit and another smaller subunit. So it is present in two forms, one larger subunit, okay, and another smaller subunit. This is larger. I'll make it a little smaller because I made it bigger than this. So I'm going to make it again. By this way, it is present in the cytoplasm. That means in the cytoplasm, the ribosomes are present in the form of two subunits, and they remain separate. But at the time of translation, both of them, they attach with each other. And this attachment of both the subunits, larger and smaller subunits, takes place in the presence of magnesium. Is very important, and in the presence of magnesium ions, these subunits, they attach with each other. That means if this is larger subunit, it attaches to the smaller subunit like this, and this attachment is in the presence of magnesium ion. Now this larger and smaller subunit both participate in the function of translation, the larger subunit as well as smaller subunit. That is why when you take multivitamins or multi minerals, multivitamins, multi minerals which the doctors give, two very important minerals are there. One is magnesium, another is zinc. Zinc is also very important because it is a micro molecule, and if even little quantity is less in the body, it creates problem. It creates general weakness, and many more biochemical reactions are hampered. And same way magnesium. See, now magnesium is required so that both the subunits, they attach to each other. That means when they attach to each other, the process of translation will take place, and ultimately what is going to happen? Protein synthesis. And protein synthesis is required for growth. So if the body is deficient in magnesium, then what will not happen? That means if the body is deficient in magnesium, this attachment of both the subunits of ribosomes will not take place, and as a result, protein will not be synthesized. So growth will not take place. That is why the multivitamin, the multiminerals and multivitamins in that these micromolecules are always present, because they are very important. So magnesium, indirectly, if we say magnesium helps in the growth of the body, of course, of the muscles also, the growth, because muscles are made up of proteins. So, and one thing more, one needs to keep in mind that these are micromolecules and these are required in very, very minute quantity. Even a little rise above the normal level in these minerals, they can lead to toxicity of that particular mineral, which may have severe side effects. So that also needs to be kept in mind, that these micromolecules, which are required, very important for the body, should not be taken in large amount. Okay, okay. So this is important. Now, what happens at these two subunits? They unite at the time of protein synthesis. Now, both the subunits, whether it is larger or smaller, they have different functions. What is the function of smaller subunit and what is the function of larger subunit? This is the larger subunit and this is the smaller subunit. The function of smaller subunit is to attach with the messenger RNA. So that means if here the messenger RNA, which is bringing the information for protein synthesis, attaches here, that means all the bases will be here in the complete messenger RNA. So first it attaches from here and then it keeps on shifting towards this side. That means once these three are read by this and this is brought, these will, suppose this is AUG. AUG always is the initiating codon. So it starts with AUG. So once it is read, it will shift more. It will shift here. AUG will come here and the next one will shift here. So this way it keeps on shifting from this here to here, and on the basis of whatever triplet codon is present here. Here the what is brought? The transfer RNAs are brought. Now, one more very important thing. The larger subunit, it has three sides. One side, second side, and the third side. This is E side, this is P side, and this is A side. The three sides are present on the larger subunit, E, P, A. A is the acceptor site, P is the peptidyl site, and E is the exit site. So acceptor, and exit. So there are three sites which are present: acceptor, peptidyl, and exit site. Now what happens? Acceptor site means it will receive, the acceptor site will receive the transfer RNA with an amino acid. When the transfer RNA brings the amino acid, it will be received by A site. Okay, and then from here, see, now this is also one thing. The initiator codon, always in the messenger RNA, the initiator codon is AUG. You all know this. This is the start codon or initiator codon. And rest we will have different type of codons. Like here we have the initiator is always AUG. Then we have suppose CCC, UUU, GGG, AUG, AUC. This way we have many more codons, triplet codons. Now, when the translation process starts, this accepts all the transfer RNAs with amino acid, but the first initiator codon is not accepted by A site. It is not accepted by A site. The initiator codon, which is AUG, is always accepted by P site. The P site accepts only the AUG, the initiator codon, and rest other those Yes. So what I want to tell you that AUG, which is the start codon, that means it will, for this, which amino acid is there? AUG codes for methionine. AUG codes for methionine. So this is not, that means for AUG, a transfer RNA with methionine will come. For the first initiator codon, the transfer RNA, I'm just making it like this so that you understand it. A rough sketch of transfer RNA. That means with methionine will come. For this, methionine will come. And this transfer RNA, which is bringing methionine for the start codon, all the messenger RNAs, they have AUG as a start codon. It will not be accepted by A. It will be accepted by B side. So always the transfer RNA which is carrying methionine for the initiator codon will be accepted